Dear students, how are you? Today we are going to talk about a very important antibiotic, that is metronidazole, which is very commonly used with the name of Flagyl, which is its brand name or trade name, right? Metronidazole, this lecture will be in this sequence, right? First of all, we will talk about intro to the drug, then mechanism of action of the drug, then we'll move on to the mechanism of resistance, right? These three things we'll be discussing today's lecture. Later on, we'll talk about pharmacokinetics of the drug, that how the drug is taken, absorbed or distributed in the body, and how it is metabolized and excreted out of the body. And of course, very important thing, that what are the clinical correlates with its pharmacokinetics. And after that, we'll go to very important part of this discussion, that is about the microbial targets of metronidazole and clinical uses of metronidazole. There are about 12 conditions I will discuss, 12 clinical conditions in which we use the metronidazole, and 6 conditions I will discuss in clinical uses part 1, and 6 conditions I will discuss in clinical uses part 2. And in the end, we'll talk about the adverse effects of this drug, untoward effect, side effects, and especially we will focus on disulfiram like action and other precautions which we should take before prescribing this drug. Now let's start with the beginning, introduction to this drug. First of all, you must know this drug is a synthetic drug, right? Synthetic drug means that it is made in vitro. It is not derived from any biological compound. Secondly, this drug is a small molecular weight drug, small molecular weight drug. This is very important to remember, even though we will discuss its small molecular weight and its importance of the small molecular weight in detail in pharmacokinetics, but right now I will mention about 2 things only. Because it is very small molecular weight, it can easily dissolve into biological membranes and cross them very effectively. That is why when this drug is taken orally, very rapidly it will absorb from the gastrointestinal system, not very rapidly only, but it will also absorb very effectively. Again, I will repeat it, when it is taken orally, being a very small molecular weight drug, it will very rapidly and effectively absorb from the gastrointestinal system, and of course it is taken intravenously, although we will discuss this thing in the clinical uses. And another important thing, that being small molecular weight molecule, right, small molecular weight, it can very easily and effectively cross the blood-brain barrier. As it crosses the blood-brain barrier very effectively, it means it can be used in brain abscesses by the susceptible organisms as well. And third very important thing is that once this drug is taken, metronidazole is a prodrug. This is very important, prodrug. What I mean by the prodrug? Prodrug means that metronidazole is basically taken in inactive form. It is biologically not active. Once it is taken or intravenously taken, or it is taken orally, or even it is applied topically, basically it is inactive compound, right? But in the biological system, in vivo, it is activated, right? And where it is exactly activated, this drug is converted into active compound, or it is activated only in the susceptible organisms. Again, it's worth repeating. Metronidazole is basically inactive compound, and there are only anaerobic organism which effectively activate this drug. So once this drug enters into anaerobic compound, anaerobic microbes, right, anaerobic microbes, inside anaerobic microbes this drug is activated. I will explain later exactly how it is activated, but you need to remember that microbes which are aerobic, they cannot activate this drug. Even our own cells, right, they cannot activate this drug. Being a small molecular weight substance, it enters in our human cells also, in our cells also, it enters into aerobic organisms also, it enters into anaerobic organisms also, but I will repeat it, as it is not activated into aerobic cells, it means our cells and aerobic microorganisms, because it is not activated, so it does not produce any toxicity there significantly. But as it is, anaerobic organisms very effectively convert this inactive molecule into active dangerous molecules, so it produces its toxic effect, killing effect, cidal effect inside the mainly anaerobic organisms. Right, now let's go into detail that what are the group of anaerobic organisms, right, which can activate it, and then it becomes produces this substance metronidazole. Once it is inside those anaerobic organisms, and after it is activation, it converts into very reactive metabolites and it damages those cells' DNA and kills them. It's a cidal drug. It is bactericidal as well as kills certain protozoa also. Let's come to this, that anaerobic microbes, right? It does not work on aerobic microbes, right? But because it is a very small molecular weight compound, it does enter into aerobic organisms also. It does enter into our cells also, because our cells also use aerobic pathway. It cannot activate this compound. In the same way, aerobic microbe cannot activate this. I will again stress, mainly these are the organisms or microbes which are having anaerobic respiratory mechanism, right? They can activate metronidazole into active compounds, right? And that is why anaerobic bacteria and protozoa are more susceptible to this drug, right? Now, anaerobic microbe, when we talk about, we will talk about anaerobic bacteria, right? And it can also talk about anaerobic protozoa. Mainly anaerobic bacteria, in which, right, there will be obligatory anaerobe, strict anaerobe, obligatory anaerobic or strict anaerobe. For example, I will just give few examples here, like fusobacterium. In the same way, I will mention about Bacteroides species. Then I will mention about Clostridium difficile, right? Clostridium difficile. So these organisms like Fusobacterium, or it is not Facebook, Fusobacterium, or I'm saying Bacteroid species, right? Or I'm saying, for example, Clostridium, what is this? Difficile, right? Now these are some of the obligatory anaerobe, right? So it can attack them, but I will tell you later that many other organisms also, when I will go into detail of what is this microbial targets. And then there are microaerophilic organisms, or there are organisms like facultative anaerobes. And when we talk about facultative anaerobes, we will specially mention two organisms where metronidazole is very effective. Number one, in the stomach, you must remember organism which is very commonly involved in production of gastritis, right? Helicobacter pylori, and then another organism in vagina, that is Gardnerella vaginalis. Now, so what I'm trying to tell you, that out of the bacteria, it does not damage all bacteria. It damages only those bacteria which are either strictly anaerobic or facultative anaerobic, because this drug can only be activated in these two type of microbes. Then if we go to the protozoa, yes, some protozoa which also have similar anaerobic pathway to produce the energy, they can also activate this drug and then they suffer, right? Especially notable bacteria, protozoa, which I should mention. Number one, there is, yes, Giardia lamblia, which produces diarrhea. We will again discuss in detail in clinical uses. Then there is your friend Entamoeba histolytica. Every medical student knows, doctor knows, which causes amoebiasis, dysentery, and sometimes amoebic abscesses. And Then there is Trichomonas vaginalis, right? So we also can remember it with the name of Protozoa get. Get means Giardia, Entamoeba histolytica, and what is this? Trichomonas, right? Now, these organisms, right, Giardia, Entamoeba histolytica, and Trichomonas vaginalis, they are also having anaerobic pathway to produce the energy, right? So, as we will see very soon that in the anaerobic pathway, right, electrons are released in such a way and captured by metronidazole that metronidazole becomes active and produces toxic or free radicals within these organisms and kills them, right? So now I will go into detail. Up to now it is very clear to you that when we talk about metronidazole, right, when we talk about metronidazole, Flagyl, the drug is inactive. Once it enters in our body, it is activated. Exactly where it is activated, it is not activated in our cells because our cells do not have anaerobic metabolism. It is not activated in aerobic microbes. Actually, it is activated in anaerobic microbes, anaerobic bacteria, anaerobic protozoa, and there, after activation, this convert into highly reactive radicals and damages the DNA and kills the cell. Now I will go into detail of, right, what happens in a cell which is anaerobic cell. Let's suppose we talk about any bacteria, Clostridium difficile bacteria. I will draw that bacteria and I will show how this drug enters into that bacterium and how it is activated and how it produces highly toxic and reactive metabolites which damage the bacteria and kill the bacteria, right? So we'll talk about this. Right, here I have drawn one anaerobic microbe, right? So we have to see how metronidazole enters into it, how it is activated into highly reactive and dangerous intermediate compounds, and how those compounds, dangerous free radicals, they react with the DNA and protein of the cell and damage it and kill this cell, right? So we have to see how metronidazole enters, how it is activated into highly toxic and reactive radicals, and how those radicals kill this cell. Before I go into that detail, let me show you the chemical structure of metronidazole very briefly, right? Actually, in metronidazole, there is imidazole ring. Imidazole ring has one nitrogen here and one nitrogen here, right? Suppose there is some side chains. So let's suppose this is carbon number 1, 2, 3, 4 and carbon number 5. Now at carbon number 1 and carbon number 3 in the imidazole ring, right, there are nitrogen, and there is a special thing about the carbon number 5, that at carbon number 5 there is a very important attachment, and attachment here is, yes, nitro group. What is this? Nitro group. So because at carbon number 5 it has nitro group, so this compound chemically is called, right, 5 nitro imidazole. 5 carbon number 5 in the imidazole ring, right, having nitro group here, 5 nitro imidazole, right? This is basically metronidazole. Now what I'm going to do for simplicity, right, and for a little fun, let me represent this imidazole ring as of the metro as a metro, right? You know, for transportation we use metro in some cities. Here is the metro, and this is representing, basically this metro is representing our imidazole ring, and here is what? Here is our nitro group. This is our nitro group. So this nitro group onward, we can imagine that this whole compound is metronidazole. This nitro group, as we will discuss, it acts as a electron machine gun. It acts like a electron gun. This nitro group actually will produce highly, when it is activated. How it is activated? Once it takes up an electron. When it takes up an electron, it becomes highly reactive, nitro group. It converts into highly reactive, you can say, radicals, and damages the cell. Now onward, this whole metronidazole is represented by this structure, right? This is imidazole ring and this is 5-nitro component, and we can also call it electron gun. What is this? Electron gun, because it should be loaded with electron, only then it will fire and produce dangerous, what? Dangerous intermediate reactive compounds which will damage the cell. So let's go into detail. Now this metronidazole, it can very easily enter into all cells. First of all, you must know, metro enters all cells, right? This is nitro, 5-nitro group, right? It enters into all cells. It can enter into our human body cells. It can enter into aerobic bacteria. It can enter into anaerobic bacteria. Why it enters into all cells? Same thing I told you, that it is a very small molecular weight, right? So it very easily dissolves into cell membranes and passes through them, and from extracellular environment, metronidazole enter into intracellular environment. But there is a very important thing. Once it enters inside the cell, maybe bacterial cell or human cell or any kind of cell, it is not dangerous until it is not activated. Again, metronidazole, once it enters into cell, right, initially it is not dangerous, right? Right? It will only become dangerous once its electron gun or nitro group is loaded with electrons, right? From somewhere it should get electrons. If nitro group does not get electron, it means it does not get reduced. If it does not get reduced, then electron gun is not loaded with electron, nitro group is not loaded with electron, so metronidazole will not be, what is this? Active, will not be dangerous, right? Now, what we need to remember, only anaerobic microbes like anaerobic bacteria or anaerobic protozoa, only they transport electron in such a way that electron can be snatched by the nitro group of metronidazole, and metronidazole become reactive, convert into reactive compounds. But electron which are going through electron transport chain in aerobic organisms, like in our cells, right, aerobic respiration, or in those microbes which are aerobic, their electron move in such a pathway that nitro group of metronidazole cannot snatch the electron. Because it cannot snatch the electron, it cannot be activated. Let's come, but in anaerobic organism, we see from where it gets the electron and how it becomes activated. Let's go for little backward in the metabolism. You know, energy metabolism, pyruvate is very important, pyruvate compound. In energy metabolism, pyruvate can be converted or should be converted into acetyl CoA, right? In energy metabolism, pyruvates is converted into process of breakdown that bigger compounds to smaller compounds, and pyruvate, which is 3 carbon unit, convert into 2 carbon unit acetyl CoA. Now, during this process, when pyruvate convert into acetyl CoA, In anaerobic organism, there is a very special type of enzyme here. This is a very special type of enzyme here, and this enzyme helps the pyruvate to convert into acetyl-CoA, right, while removing electrons, and electrons are transferred, right, to a electron carrying compound, that is, or electron ferredoxin. Ferredoxin is a special electron transporter protein. Ferredoxin is a special electron transporter protein which is present in anaerobic microbes, anaerobic bacteria, anaerobic protozoa, right? Now, this ferredoxin, right, with the help of this specific enzyme, right, act into this pathway and, right, gains electron. Ferredoxin gains electron. I mean, I will show here that ferredoxin is gaining electron, right? And this is ferredoxin. Now, here it was without electron, so it was in oxidized form, and here it converts into electron loaded state, it gets into reduced, right? So this process in which pyruvate convert into acetyl-CoA, there is a special enzyme which help the ferredoxin to interact with in this pathway, and the electron which is related with the pyruvate pathway to acetyl-CoA pathway, those electrons are transferred to the ferredoxin. It means in this situation, ferredoxin is getting reduced. Ferredoxin is getting reduced, right? Now, this is oxidized form of ferredoxin. This is reduced form of ferredoxin, right? I think let's go to very basic. You must be knowing a concept of a mnemonic oil rig. This is a mnemonic to remember that oxidation occurs when there is loss of electron and reduction occurs when there is gain of electron. In this metabolic pathway, what is happening, that ferredoxin, right, is gaining electron, so it is getting reduced, and pyruvate is losing electron while converting into acetyl-CoA. So pyruvate is getting, what is this, in which form? It is losing electron, right, to ferredoxin, and ferredoxin is electron loaded. So this activity is done by this enzyme, and this enzyme is called, yes, pyruvate, ferredoxin oxidoreductase. What is the name of this enzyme? I will write it here. Pyruvate, yes, ferredoxin oxidoreductase, right? So this P4, I will just call it P4, but actually its full name is pyruvate ferredoxin oxidoreductase. This enzyme is responsible to convert the pyruvate into acetyl-CoA and convert the ferredoxin from oxidized state to the reduced state. Now we have seen that this specific enzyme is present in anaerobic microbes. But in aerobic microbes or our cells, which are aerobic cells, this pathway, pyruvate to acetyl-CoA, is done by another enzyme, and that enzyme is called pyruvate dehydrogenase. In our cells or in aerobic organisms, this conversion of pyruvate into acetyl-CoA is assisted by enzyme pyruvate dehydrogenase, right? Now, because in our cells we are having different enzyme, anaerobic bacteria do not have pyruvate dehydrogenase, so they use pyruvate, what is this, ferredoxin oxidoreductase. Now here is the classical thing. When the in this process, as energy metabolism is progressing continuously, it is converting cycling oxidase form of ferredoxin into electron loaded ferredoxin, which is reduced form of ferredoxin. Reduced form of ferredoxin, from it electron can be very easily snatched into what? What is this? Yes, this point is very, very important, that from electron loaded ferredoxin, we can also call it reduced form of ferredoxin, right? Metronidazole will interact with ferredoxin and snatch away or grab away or suck away its electron. So electron will be transferred to the nitro group, right? It means now, what is this? Metronidazole. Metronidazole, by receiving the electron, will convert into what? Reduced form. And in reduced form, when it is loaded with electron, right, this nitro group will convert into electron gun, which is fully loaded, and now it will fire highly reactive, what is this? Intermediates, right, and kill the cell. So we can say that this is what kind of activation? What is this kind of activation? We call it reductive activation. In simple way, you can say metronidazole enter into cell and electron is added from the ferredoxin, and metronidazole nitro group, when receive electron, it will become highly reactive, right? So now we go over here, that this metronidazole, which is loaded with what? Electrons, or it is now into which form? Reduced form, right? In reduced form, it is highly reactive, and we can say because it is having electrons, it is highly reactive, and it is going to fire, what is this? Highly toxic compounds, right? Intermediate toxic compounds. Now what are those compounds? Let me show you. Okay, before I go for those compounds, I want to tell one thing. This is, what is this? Inactive metronidazole, right? Inactive metronidazole which enter into cell, and it is activated by a reductive process by gaining the electron. It receives the electron from ferredoxin, which is an electron transport protein, ferredoxin. It receives the electron from reduced form of ferredoxin, and its nitro group is activated. Is that right? And now here is activated metronidazole, right? I will see this activated drug, the pro drug has been converted into active drug within the anaerobic cells, right? And there it will produce highly toxic compounds. But remember, as pyruvate to acetyl-CoA transformation is continuing for energy production, right? So ferredoxin keep on converting into reduced form, right? And electron keep on sinking. Electron keep on sinking into electron sink. Electron sink is our electron gun. That is the nitro group of metronidazole. So metronidazole from inactive convert into active form. As more and more inactive metronidazole is converted into active form, from diffusion, more inactive, what is this? Metronidazole molecule will keep on entering into this cell. So what we can say, the more ferredoxin donates the electron to metronidazole molecule, more metronidazole molecules become activated and dangerous, right? And more inactive form keep on diffusing into cell, right? But remember, once this molecule enters into cell, it receives the electron or it undergoes reductive activation. Then because it is highly charged molecule, right, receiving the electron, right, so it is trapped within the microbe. It cannot leave the microbe now. Why? Because it is heavily charged, right, and it is charged molecule, it cannot dissolve into membrane, so it cannot leave out. So what really happens, that anaerobic organisms, right, they keep on doing activation of the metronidazole by donating the electrons from ferredoxin, right? And more and more metronidazole molecules convert into highly reactive compounds, and highly charged and highly reactive compounds are trapped into cell. Then they will start damaging the DNA and other structures. And as more and more is being activated, right, more and more inactive molecules will enter, right? But in those cells which are aerobic, they do not have pyruvate, ferredoxin, oxidoreductase. They don't have this enzyme, and they don't have ferredoxin, aerobic molecule, right? They have other enzyme in aerobic, microbes, aerobic microbes. They are having not this enzyme, they are having pyruvate dehydrogenase, aerobic organisms. And due to pyruvate dehydrogenase, right, this pathway which is present in aerobic, right, the electron transport chain in the aerobic molecule, right, aerobic microbe, they hold the electron in such a way that nitro group cannot snatch the electrons, because in aerobic organism electron transporter chains hold the electron in such a way that they don't give electrons to the metronidazole. So metronidazole is not activated in the aerobic cells, human cells or aerobic microorganisms. Now we come to that. So this was anaerobic cell, and this very unfortunate anaerobic cell which was unfortunately having ferredoxin, unfortunately having, what is this? Pyruvate, ferredoxin, oxidoreductase, and very super unfortunately received the metronidazole molecule, and then it gave it 2 electrons, and this become activated into in an anaerobic organism, and now this will produce highly reactive compound. What are those compounds? Let me show you very briefly here. That let's suppose these are imidazole ring. This is imidazole ring, and here is nitro group, right? And this nitro group, this is oxygen, and this is also oxygen, right? Now this is the normal, what is this? Metronidazole, which is inactive. But once the electron is added here, this electron is added here, this will convert into, this compound will convert into reduced form, and in this reduced form, yes, it will go into this form. Okay, I will show you the electron here and electron here. Now this is highly reactive, this is highly reactive compound, dangerous compound, right? This is actually, which is loaded with electron, it is called nitroso free radical. What we call it? Nitroso free radical. It is highly reactive. It will react with DNA of the cell and react with the proteins of the cells and damage them. Then if it receives hydrogen, it will go into further change. This will go into further change and convert into, let me show exactly, another reactive compound. Okay, NO. It will go into this form, and then it will go further change, right? I will show you this, and here it has released one superoxide. Now this superoxide, which is released in this process, when it is try to stabilize itself, nitrogen with oxygen, one double bond and one superoxide is released. This superoxide is also what? Highly toxic or reactive compound. We call it oxygen free radicals, right? So basically nitroso free radicals are there, superoxides are released also there, and we see another compound which is going to be dangerous in the cell. If some hydrogen is added here, right? So what will happen that this nitrogen and this O will receive OH and hydroxyl group. That is also highly reactive. So what we see, there are three highly reactive compounds that once electron is added here, this was our electron gun, when it gets loaded, first of all it converts into nitroso free radical, which is highly dangerous. Then it converts into a compound which will release, what is this? Superoxide, which is highly toxic. Then it can convert into a compound which is having hydroxyl reactive group, that is also highly toxic in the cell. So we can say that metronidazole enters into cell into inactive form, but once it receives the electron from reduced ferredoxin, metronidazole itself gets reduced, and reduced metronidazole is an activated, what is this? Compound, and this compound at nitro group, after its reduction, nitro group convert into nitroso free radical, then further changes occur, it will release superoxide ions, further changes can occur, and it may have intermediate compound with hydroxyl, right? And this hydroxyl is also reactive. So there are different ways to say. If you cannot remember all this, no problem. You just remember this, that metronidazole after reductive activation convert into highly reactive toxic intermediates. Highly reactive toxic intermediates. Or another way to express the whole process is that once metronidazole receives the electron and get reduced form of electron produces, right, oxygen free radicals, right? And these oxygen free radicals or reactive oxygen species, there are three names for these situations. Number one, highly reactive, what is this? Intermediates, or we call it nitroso radical, superoxide and intermediate with hydroxyl end, or we as a group can call it highly reactive, what is this? We call it ROS. As a group we can call it ROS. ROS means reactive oxygen species which are part of these molecules. So these are highly toxic molecules. They will react with DNA of the cell. They will react with the DNA of the cell, right? And they will uncoil the DNA, or even at many points they will break the DNA, damage the DNA. And you know very well, if DNA of the cell is damaged, right, this anaerobic cell, when DNA is damaged, it cannot undergo replication process. It cannot undergo, what is this, from DNA to RNA formation for the protein, which is called transcription process. Again, if DNA is damaged, you cannot, then this cell cannot make DNA from DNA, so replication fails. This cell cannot make RNA from DNA, so transcription fails. When replication and transcription fails, then proteins are unable to be synthesized, and these reactive compounds not only damage the DNA, they also react with cellular proteins and damage them also. So damage to the DNA by the reactive compounds and damage to the proteins, all this mess up with the whole intracellular molecular machinery, and cell will undergo death process, or we say this cell is now killed, or we say that metronidazole, once it is activated, it has acted as a bactericidal compound. In highly reactive form and reductive form, metronidazole is bactericidal compound. Is that clear? And in the same way it can kill the protozoa. So whole this story is about only one thing. What is that thing? That in aerobic organism, we don't use ferredoxin for electron transport, right? As they don't, aerobic organism in their cells, because they don't have reduced ferredoxin, they cannot activate the metronidazole. So metronidazole cannot get activated into aerobic cells and cannot produce highly toxic or reactive compounds and cannot damage aerobic bacteria or our cells. Again, in a nutshell, in anaerobic bacteria, there is lot of ferredoxin in reduced form available, right? And this keep on giving its electron to the nitro group of metronidazole. This nitro group act as a electron sink or electron snatcher or electron grabber or electron sucker, and once it is having the electron, it becomes highly reactive, right, and damage the cell and kill the cell. So this is clear up to now. Now we go to the next point, that if oxygen comes to this system, let's suppose hypothetically, if you introduce oxygen into this system, right, oxygen will take up the electron from here, and if electron are taken up by the oxygen, these compounds will be recycled and become inactivated. That is why anaerobic organism having metronidazole in anaerobic environment will be killed. But if there is anaerobic bacteria and we supply a lot of oxygen into that tissue, and if that oxygen take up these electrons from here, then they will become inactivated, right? Now we come to this thing, that susceptible organism, which are anaerobic organism, in some situations they become resistant to metronidazole. So now we are going to talk about mechanism of resistance in metronidazole. Here I want to say the good news is that anaerobic bacteria have resistance to the metronidazole very rarely. What I'm saying? Rarely. So anaerobic bacteria, right, because they are rarely resistant to the metronidazole, so we say still metronidazole is very effective against the anaerobic bacteria, right? Even though some resistance is increasing in some countries like India, I will tell you about the mechanism. Then, but when we talk about resistance to the metronidazole, we should remember two microorganisms. Number one, Helicobacter pylori, right? About in some cases, in some studies, up to 30% of the Helicobacter pylori isolates are found to be resistant to the metronidazole. And another organism in which resistance to the metronidazole is relatively common, that is Trichomonas vaginalis. So if you have to remember about the resistance, right, you have to remember anaerobic organism resistance is very rare against the, what is this, metronidazole, but resistance against metronidazole is commonly seen in Helicobacter pylori and what is that, Trichomonas vaginalis. Now what is the mechanism of resistance? Most common mechanism of resistance is, that either these cells have reduced, they have reduced activity of pyruvate, ferredoxin, oxidoreductase. For example, Helicobacter pylori or Trichomonas vaginalis, right, which are sometimes resistant, the resistant strains will have reduced activity of PFOR, right? Rather than they have another enzyme here, right, and bypass this pathway. That enzyme is called lactate dehydrogenase. Lactate dehydrogenase. If lactate dehydrogenase enzyme is increased in concentration in anaerobic organism, so pyruvate will go by other enzymatic pathway to acetyl-CoA, right, eventually, or you say not to acetyl-CoA, to lactate. Again I will repeat, in anaerobic organism like Helicobacter pylori and Trichomonas vaginalis, if lactate dehydrogenase expression is increased and expression of pyruvate, ferredoxin, oxidoreductase is decreased, then pyruvate is giving electron to other pathway which is mediated by the lactate dehydrogenase, and pyruvate converting is not giving electron and converting the ferredoxin into reduced form. right? And if reduced form of ferredoxin is not available, then of course metronidazole, which is entering into this resistant microbe, this metronidazole will not get, what is this, undergo oxidative reduction, will not get the electrons and will not produce the highly toxic or reactive intermediates, and cell will not be damaged, right? So main mechanism, most common mechanism in Helicobacter pylori or Trichomina vaginalis or any other anaerobic organism which become resistant to the, what is this, metronidazole, most commonly what we see that actually reduced ferredoxin is reduced form of ferredoxin is less due to reduced activity of, what is this, pyruvate, ferredoxin, oxidoreductase, and there might be increased activity of lactate dehydrogenase, right? So pyruvate, right, pyruvate converting into energy producing compounds, it takes up other pathway rather than doing the loading the electron on ferredoxin, and as electron loaded ferredoxin is not being made in these microbes, resistant microbes, so metronidazole is unable to get the electrons, so it cannot going under reductive activation and unable to damage the cells. But another thing, once metronidazole is not activated, right, it accumulates in the cell into inactive form, do you think more inactive will come? No. So uptake of the metronidazole will be also reduced, because in anaerobic organisms which are susceptible to metronidazole, there is a very high uptake. Why this high uptake? Because whatever molecules of metronidazole enter in, they are being converted into active form, and more inactive molecules of the drug are undergoing into cell under the concentration gradient, right? So what happens? If this is less and lactate dehydrogenase is more and reductive ferredoxin is less, electron is not being provided to, what is this, metronidazole. So metronidazole stays inactive, so concentration gradient is not built and uptake of the metronidazole is reduced. So we can say one mechanism of resistance is reduced uptake, but main mechanism is reduced available of electron loaded ferredoxin, or we can say reduced, what is this, reductive activation, and in some cases there is increased efflux also, increased efflux. So it means there can be reduced uptake for resistance, there can be decreased activation, and there can be increased efflux, right? So these three mechanisms can be there for the resistance. So here we stop our today's discussion. In next discussion we will talk about the pharmacokinetics, right? And pharmacokinetics is simple. What really I want to teach you, the clinical correlates involving the pharmacokinetics, that in liver diseases or renal diseases, how do you adjust, in patient which is breastfeeding, what to do, how to decide about this drug, and then especially drug interactions which occurs because this metronidazole molecule we will discuss, it has very special interaction with cytochrome P450 system and the other drugs which also influence the cytochrome P450 system in the liver. That will be discussed in the next lecture. Stay tuned. Best of luck.